Welcome back. And so today we're going to continue on with the concept of mapping. So in chapter 5, we looked at mapping of chromosomes a little bit, and we saw a lot of, we looked at the concept of crossover and how genes are recombined in chromosomes by exchanging material, by exchanging genetic material or pieces of the chromosomes. So today we're looking at the mapping in bacteria and how crossover or the equivalent of crossover in bacteria occurs. And we'll look at bacteriophages also. We'll get to definitions of those soon. But we're continuing on with this because the bacteria many bacteria like E. coli, they exhibit the characteristics of a model organism that, and we looked at those parameters or what constitutes a model organism in chapter 1, I think it was, where it's got to be easy to use and fast growing and has a lot of offspring. And so that's what E. coli it basically exhibits. It's considered the workhorse of biotech labs and comes from our gut. Some of it can be deadly, but there's a lot of strains that are not, that actually are very good hosts for putting DNA into and letting it grow. Or recombining the DNA, and we'll see some of that in this lecture here. So this lecture actually I didn't put an overview. I didn't put our learning goals in this chapter, but it's pretty straightforward. I think section 6.9 is the only one that you really don't have to take a look at, but I'll put more detail on that on Canvas. So here basically, by way of introduction again, we saw in the last chapter that genetic mapping was done in the fruit flies, and it was done extensively in fruit flies, but in humans it was a lot more difficult. And even though chapter 5 didn't mention the computer applications for sequencing the genome, that's really where we are today. So as nice as it is to know about the extensive mapping in the fruit fly, that would be really pertinent if you were going into a research environment, if your goal was to work in not just genetic research, but really any kind of biological research, where fruit flies might be an option. But fruit flies are kind of the organism of the past. Nowadays, there's so many other organisms that are great to work with, like yeast is one, E. coli is another. There's so many different ones out there that something like fruit flies that's difficult to count and difficult to maintain any control over. That's an organism of the past, really. So. But still, it's nice to see that because it's nice to see the sequencing there and to understand it because it does still apply to a lot of model organisms. So again, the benefits of using any microorganism, whether it's a fruit fly or a virus or a bacteria, the benefits are that here in particular, bacteria and bacteriophages are very much essential to genetic study. They often act as hosts for putting segments of DNA, inserting segments of DNA into it, and then watching what happens under a variety of different circumstances or environmental conditions. Bacteria, though, in particular, has developed a lot, many strains of bacteria have developed ways of exchanging and recombining genetic information between cells. So, if you study that, if you look at that, you can learn a lot about how human cells behave. Oftentimes, there's not that much difference between looking at bacteria or looking at a segment of human DNA inserted into a bacteria as the host to see what would happen. So bacteria it gives us a lot of important information about recombining DNA or recombining genetic information between cells as well as within cells. And bacteria provides, or looking at this information or the exchange of information, it gave us the basis for mapping the bacterial genes. So watching and understanding how genes were exchanging genetic information or within bacteria, how the bacterial cells or bacterial genes were exchanging that information, it gave us a way to gave geneticists a way to figure out what the mapping of the genes, of the bacterial genes are. So both bacteria and viruses are very valuable in research. They have that very short reproductive cycles, and they can be studied in pure culture also. So even though they reproduce rapidly, like the fruit fly, they also have a short reproductive cycle. So you much like the pea plant with Mendel, where you can grow a whole bunch of generations or do a whole lot within one season. So the same thing is true of bacteria and viruses. And then studying in a pure culture, you don't really have to make a lot. Often an agar solution is enough to put bacteria in and a virus in, and then observe what happens or do what you need to do. It's considered a minimum nutrient broth or a minimum amount of nutrition to keep the virus or the bacteria alive and growing. So it's often considered a pure culture because you often also don't have to mix it with anything else either. So some of the ways of understanding how our bacteria recombines. So understanding how bacteria recombines or strains recombine, we first need to really understand mutations here. So a mutation is a change. It's something that's beyond the normal or a change from the normal. So genetic studies with bacteria, a lot of the original purposes were to study mutations in those organisms. And homogeneous cultures often give rise to cells that show variations. A heritable variation is a mutation that gets passed from generation to generation. And those variations can occur, or we can see those variations happening, popping up, giving new rise, I should say, to new cells with heritable conditions, variations. When these cultures are grown in unique environmental conditions, so that's really another way of saying a very controlled environmental condition. You can have one environmental condition at one temperature and another condition at a different temperature, and have the same culture in each environment, and see how many mutations may occur, how many heritable variations end up resulting from those changes. One example is E. coli, and we're going to see E. coli a lot in this chapter. It's susceptible to infection by the virus, with the bacteriophage T1. So the virus is actually going to reproduce or spread all the while it lyses its host, the E. coli host. So the virus, the bacteriophage is a virus, and the T1 is the name of a particular bacteriophage. And we have a virus called the bacteriophage, and that bacteriophage is named T1. And this T1 virus, or this virus, it spreads and reproduces all the while it kills off its host cell. So E. coli is very susceptible to that infection, and so being susceptible to it means it can't withstand the invasion. So the virus latches onto the E. coli host, and it just cuts it all up. Lyses it means it's breaking it all apart. So rare E. coli, very rarely does an E. coli cell actually survive a bacteriophage T1 attack. But however, there are those rare instances when some cells can survive. It's kind of like instead of destroying it into oblivion, you have some remnants left over. You could think of it that way. You have some E. coli cells left over in the wake of this viral attack. So those leftover cells, when they're isolated and cultured, their descendants, so when they're cultured, they're going to replicate. You're putting them in something that's going to allow it to grow and nourish it, grow it, reproduce it. So when that's done, when you take these surviving cells out of their original environment, put it in culture, nurture it, and allow them to grow and reproduce, their own descendants become resistant, or their own descendants are. It shows that those resistant, or it shows that the rare E. coli cells that survived, their descendants are resistant to E. coli infection. So that's what the adaptation hypothesis is, that there's an interaction of a bacteriophage and a bacterium, and that the interaction is essential to acquiring immunity to the phage. And exposure to the phage induces or brings about that resistance in the bacteria. So we know that concept. You're probably very familiar with immunity and vaccinations, where and as well as even aside from vaccinations, acquired immunity is something that you have to build up. Your body needs to build up that type of immunity to resist some type of invasion or some type of viral infection, and you could acquire it by being exposed to it in a variety of different ways. So in this case here, the exposure of the phage to the bacterium brings about a resistance to that infection in the bacteria. But another way mutations can happen. So the last slide, The last slide here, that's a good mutation. We have this E. coli surviving cells, or that had been exposed to an infection and survived. So a mutation was that, or an induced mutation was that resistance to the bacteria. So another mutation it's not an adaptive mutation, rather it's a spontaneous mutation. And that's the spontaneous mutation is actually considered the primary source of genetic variation within a bacteria. So it occurs in the presence or the absence of the bacteriophage T1. So again, a bacteriophage is a virus that infects bacteria. So a spontaneously arising mutation can occur when you're looking at something in culture. And when you see that mutation arise, you can isolate it and establish it in its own culture, independent of the parent strain, and then let it grow, let it see what happens. And oftentimes we see selection occurring where the cultured mutant strain is under certain conditions so that it only grows well. It's not put in any kind of adverse condition that could be harmful. It allows it to continue to grow. So we're basically helping to select the fittest among them. We're not really putting it in extreme circumstances. So we're able to select this mutation and grow another strain or grow another strand of genetic information, whatever it may be that we're looking at. We can isolate it and end up inducing another strain that's going to grow well. So the selection part is, again, the growth of an organism under certain conditions, very good conditions that allow for that strain to prosper. We only want that desired mutant to grow and grow well. The wild type does not. We're not mixing and matching. We're only looking at that mutant here. We know the wild type proliferates. We already know that that's prospering and creating offspring. But in this case here, we found a mutation, a spontaneously occurring mutation, and we want that mutation to thrive and grow. We want to be able to do that because there may be other strains. We may find some extra research benefits of it. We might find some applications to human chromosomal adaptations or mutations within our own DNA. So here in this case, the ability to select a mutation and isolate it and then let it grow often has a lot of very good benefits within genetics research. And when we're able, it's got to be a mutant though that is able to be isolated. If you isolate a mutant and you put it in a culture and you can't do anything with it, there's really no point in moving, in trying anything else with that. Here we want to select the fittest ones. We want to put mutants, isolate a mutant and put it in a culture in certain thriving conditions. And if it doesn't thrive in there, there's no sense in keeping it in that way. You can try a different condition, and maybe it doesn't work well in cold, but maybe in a warm environment the mutant would thrive. So you could change the conditions and try that way to grow this new mutation. Typically, bacteria and viruses only contain one copy of a single chromosome. So all mutations are expressed in the descendants of that mutant cell. It's just because it has just that single chromosome. When the mutation occurs in that chromosome, that mutation is going to always be expressed in all the descendants. So that's definitely different than humans, obviously, where one chromosome can have the mutant and it can be recessive, so it doesn't show up. But here, bacteria and viruses are often single chromosome, and you would see that mutation carried forward. And then the media that you use, the nutrient media, again, it's usually an agar plate, but semi-solid agar in a petri dish. But the bacteria are grown, they could be grown in a liquid culture medium or a semi-solid agar. So the liquid medium, that's the one that's more likely or more often called a nutrient broth, just because we think of chicken broth or some kind of a vegetable broth or something like that as a more of a fluid or liquid. So here, the liquid culture would be more like a broth, whereas the agar is more gelatinous, semi-solid. So, but when you think semi-solid, don't think of a lot of water in that. It's just a really flexible solid is really what that means. So if you're familiar with gel electrophoresis, that gel is not real rigid. It's very gelatinous. Obviously, it's called a gel. Well, that's really what a semi-solid is going to look like here. These semi-solid agar dishes, you can pierce them easily with a toothpick. You can do and they're made that way because maybe you want to take your cell instead of sitting the cell on top or your specimen, whether it's viral cells or bacterial cells, instead of just sitting them on top. Maybe you want to slice that agar open and put the cells inside, surrounded by the agar. So that's why you would really want a semi-solid mechanism instead of something that's completely hard. But maybe liquid it allows it to disseminate or break down more. So that's maybe the liquid's not the best solution. So you get somewhere in between the liquid and the solid. And then your medium can have your minimal medium is it's just got the bare basic nutritional components, and those they're very simple components. It could be one organic carbon source or an inorganic ion, typically in there that's going to feed your specimen. But when you have a complete medium, that's where the amino acids are all added as a supplement to the minimal media. So you're putting a whole lot of food here, and you would want to do that when your specimen cannot synthesize all the necessary compounds. So when that's the case, which is usually the case with a mutant, you would want to add some amino acids that it could take up and synthesize. So a couple of definitions here: prototrophs and autotrophs. So the prototroph is the wild type or the common, and that can always grow on the minimal media because basically the prototroph is self-sustaining, and it's able to synthesize all the organic compounds that it needs based off that minimal amount of nutrition. But the mutant is the auxotroph, and it cannot synthesize essential organic compounds any longer. The mutation rendered that capability obsolete. It got rid of that capability. So the auxotroph lost the ability through the mutation to synthesize the essential compounds, and consequently it needs that complete medium in order to grow. So several growth phases that you often see. So we have a petri dish here, an agar plate with a medium. It's just by looking at it, you really can't tell that agar's in there. It's just more often than not that seems to be the material of choice in lining your petri dish. So here we have three different plates with three different growth phases. So we have the lag phase is slow growth. The log phase would be really exponential or a whole lot of growth, and so that would be this would be the log. This one here is your log phase or lag phase. I'm sorry. Let me start that. Let me write that again. So this one's your log phase. I said that the first time because you got exponential growth there. This is the lag phase. You have slow, and this is the stationary phase. So they're actually pretty watered down though. So I think the creation of these dishes meant that there was a lot of fluid that was added added to them, or the dilution steps actually that in the culturing of this bacteria. So each dilution was done by a factor of 10, and then each colony was derived from a single bacterial cell. So all of these individual dots really are colonies, but we can see here that there's a cluster right there of colonies, clusters, so they clump together. And then obviously here in this dish, pretty much everything, it's hard to tell the single cell or the single bacterial cell any longer, where once they began growing, it just got so prolific that each individual cell ended up sticking with other cells, and you got all these little groupings. We can see right there just one little single cell, and same thing right there. But for the most part, they were proliferating and growing all around them. So that genetic recombination occurs in bacteria. We know that now, and it's similar to crossing over. So with the chromosomes, the crossover, if you think of chromosome 1 and chromosome 2, well, if 1 and 2, these right there, the sister chromatids would recombine, so you can get a little bit of you get that crossover here, so they exchange the genetic material. So instead of having a blue and a red, we now have a combination of each. So they exchange that genetic material, and it could be this way, or it could be this way here. It could be any different combinations, to exchange that material. So within a bacteria, the bacteria genetically recombine in a way that's similar to crossing over, but it's different though. The methodology, the recombination, provided the basis for the development of chromosome mapping methodology. So genetic recombination with respect to, that's what WRT stands for, with respect to. So genetic recombination with respect to bacteria means that the replacement of one or more genes present in the chromosome of one cell is going to get replaced with those from a chromosome of a genetically distinct cell. So looking at that again, genetic recombination is the replacement of one or more genes present in the chromosome of one cell with those from another chromosome, a genetically distinct cell in another chromosome. And it differs from crossing over in the sense that crossing over gives you a reciprocal change of information. So we saw here these chromosomes here, when they crossed over, it was reciprocal. Each one of these got a piece of the other. But in genetic recombination, there's no reciprocal exchange. You're just taking... sorry about that. So in crossing over, you're taking these pieces of material from the chromosomes. It's a reciprocal exchange of material. But in genetic recombination, it's not an exchange. You're just taking, you're replacing one or more genes in the chromosome of one cell with those from the chromosomes of a chromosome of a genetically distinct cell. So you have two cells here, and you have we'll put some little blue material right there. So maybe you come over and you slice that in half right there, and you take that little tiny fragment and you put it in there. So it's just really replacing of one or more genes present in the chromosome of one cell with those from the chromosomes of a genetically distinct cell, or genetically different, a distinctly different cell. So it can't be, if you have a cell, like when you think of the daughter cells divide in there, they're not distinctly different yet. We're talking about two cells right there, and we're replacing the genes present in one cell with those from the other cell. But the other cell, the host cell, the one that's giving up its cells, nothing happens. It doesn't gain anything. It's just losing some of its genetic material there. So the genetic information is transferred in both crossing over and genetic recombination. So genetic information right here, when we do this crossover, genetic information is transferred from between the two, even though it's a reciprocal. Even though it's a reciprocal exchange, we're still transferring genetic information between those chromosomes. And similarly here, between these two, we're transferring genetic information from one to the other. Both crossing over and genetic recombination, both of those result in an altered genotype. So that transfer of genetic information from one cell to another, there's two different types of that transfer. The first is vertical gene transfer. So it's transfer of genetic information between members of the same species. So again, same species. You can't have two different species vertically transferring genetic information. So if it's a vertical transfer, the transfer is occurring between the same species. But when it's a horizontal gene transfer, the genetic information gets transferred between related but distinct species. So bacteria is one of them. You can have different types of bacteria, and they're related. They're still in the family of bacteria, and but they're different strains or different types of bacteria. And but between those distinctly distinct types or family of bacteria, you can transfer that genetic information. So the genes involved when this happens, when you're transferring within the same species, related but distinct species. They're again. I'm sorry. I said same. Same as vertical. But with horizontal, they're related but distinct species. When that transfer happens, it often plays a significant role in the evolution of that bacteria. So often the cell or the bacteria gaining the information maybe makes it stronger, a little more stable, a little easier to resist information or resist infection. So oftentimes that bacteria evolves into a better form of itself. The genes confer survival advantages to the recipient. So some examples is transferring antibiotic resistance to another species, or in some cases, enhancing the pathogenicity. That enhanced pathogenicity can also be transferred between those distinct species here. So that process, again, we have two different types and three different ways that it could happen. And conjugation, transformation, and transduction. Transduction we're not going to really get too much into in the lecture. Conjugation and transformation are the key ones because oftentimes transformation and transduction can be very similar. So oftentimes you really can't tell them apart. So we're going to take a look at each of these here. So we'll start with our conjugation. So conjugation is genetic information from one bacterium is transferred to another, and they recombine at independent locations in order to become a wild type cell, the more common cell. Prototropes when we have conjugation in bacteria, we can get prototropes to result from two auxotrophs, and we'll see a little example of that actually right here. So in this case here we have two strains, strain A and strain B, and both of them are auxotrophs or autotrophic strains, and they're growing separately in their own little containers. So we have strain A and strain B growing separately. But in those strains, when they're growing, it's really unlikely that mutations, that if you have many mutations or even three or more, it's really unlikely that the mutations would all undergo a spontaneous mutation at the same time and at all those different independent locations in order to become a wild type cell. So the auxotrophic cell is the mutant, and what we're saying is that a mutation could occur that then makes the mutant back to the prototroph. But it's really unlikely for that to occur spontaneously. So highly unlikely that the spontaneous mutation and then the recombination or spontaneous mutation is going to revert the auxotroph back to the prototroph. So the figure here shows an experiment in which that genetic recombination, or it shows an experiment in which genetic recombination occurred when the two different strains were mixed in a complete medium. So we have strain A over here, And strain A over here is growing in its own medium, and strain B over here is growing in its medium. So the negative up here means methionine and biotin are not present, but they are present over here. And then threonine, leucine, and thymine are not present there, but are present over here. So in that nutrient broth, it's a fluid here, so it would be more of a broth here, liquid medium. So they're in this liquid medium, and then you take them out. After you grow your cells, you take them out, and you plate it on a minimal medium right here. So the minimal medium doesn't have any of these, this food source, and nothing happens. You don't get any growth, and you certainly don't get any transformation from the auxotroph to the prototroph. And then the same thing's happening over here. You're growing these strains, and then you plate it and incubate it, and nothing happens. You don't get any prototrophs there either. But when you combine them here, you combine them into one, and then so that means you have methionine, biotin, threonine, leucine, and thymine all present in this container here. And then you put it on this minimal media and you incubate it. But what ends up happening is when you do that, when you mix them and then you put them on this plate, you wind up with the cells growing, and they're growing at a frequency of 1/10⁷ of the total cells. So 1/10⁷, that's still a really small amount. So it's still a rarity, but we know it does happen. And you can see here that strand A grew once it got added, or once it got access to that methionine and biotin. So it didn't have that here. It didn't have it here, and it got put on this plate and didn't grow. But when it got put on this plate, it grew because it got mixed with the right nutrients. So it got the right nutrients from strain B. And conversely, strain B got its nutrients from strain A. So they were feeding off each other, and they crossed over. And so with bacteria, crossing over is not the official term. Genetic recombination is the term. So recombination between these strains resulted in the auxotrophs becoming prototrophs. So those prototrophs can be taken out, isolated, put back into its minimal media, and grow rather well. So we saw here, that's a good example of discovering that genetic recombination occurs, or that differing strains can feed off one another. So that brings us to E. coli, which oftentimes it's self-sustaining. It's a model organism, and oftentimes it just needs that minimal media to grow. You can give it additional things and it can grow more prolific, but the minimal media is sustaining enough. E. coli chromosome is actually a circle, and the order in which the gene enters when we're recombining, when there's genetic recombination, it depends. There's a variety of different entry spots, or where the origin first is. And the first part of the donor chromosome to enter the recipient, that's where the origin ends up being established, is where that point of entry is. The E. coli model organism is used as a host for introducing. So if it's a host, you can put DNA sequences and their protein products right into that E. coli organism, and you can watch the protein products grow or change, and you can recombine or combine the DNA, genetically engineer the DNA. So there's a lot of different things you can do when you put that DNA sequence into the E. coli as a host. So research with E. coli led to determining the nature of gene replication, understanding gene organization, as well as the regulation of genes. And it also provided the evidence for the basis of mutations. Looking at putting DNA into E. coli, or just doing an experiment like we saw in the last slide. That last slide showed that when the two strains were combined, genetic information was exchanged, and then the auxotrophs reverted back to the prototroph. So that's evidence right there that mutations occur. And then also putting DNA in E. coli, it's a chance for achieving genetically modified organisms. It's a way that allows for seeds to be manipulated to make them drought resistant, maybe beef or cattle beefier, or food sources beefier, like bigger tomatoes. A lot of different things you could do with genetic engineering or genetically modified organisms to produce better food. Most people run away from GMOs, but oftentimes the food is actually better. It might be sweeter, again, drought resistant, resistant to certain insects or bugs or pollination. So a lot of good things come from it. So again, E. coli is what led to those discoveries. So here's our E. coli circular chromosome, and we have the four different strains here, and you can see the order of the genetic, the order of the the order of the transfer. Here's the origin. That's where the bacteria or the genetic information enters the E. coli chromosome, and here it's entering on this part. It's entering down here and right there. So each of these four strains have a different origination, and that origination is the entry point, the first time that the other cell or the other chromosome entered the E. coli chromosome. And then that brings us to our topic of plasmids, which are similar to E. coli, but they are different. They are distinctly different. Plasmids are double-stranded, closed circles of DNA, whereas bacteria is often one strand. And plasmids exist in multiple copies within the cytoplasm. They're naturally occurring. They're considered extras within the cytoplasm. They self-replicate the genetic elements within the cell, and they contain one or more genes. The genes help the host bacteria become resistant to antibiotics, or the genes, other genes of these genes that are contained by plasmids. Maybe some of the genes allow for digestion of unusual substances, and yet other genes kill off other bacteria. They act more as defenders of invaders. The plasmids replicate independently of the bacterial chromosomes, and plasmids, like their E. coli counterpart, they're also used in genetic engineering and to generate recombinant DNAs. So you're recombining DNAs here with by using a plasmid. And the plasmids are also used as a mechanism of transferring genes between organisms as well. So here's what a plasmid looks like. And oftentimes you want to come over and cut this part. You can take this part and you could cut this part out right there, and then insert your DNA sample. If you want to study, if you're trying to study DNA in a host organism, maybe you want to insert the DNA there, and then insert the plasmid in something else. But either way, the plasmid is what you recombine or reconnect or put that DNA into the plasmid, and then the plasmid into something else, and see how the combination survives. So plasmids have two components. The resistance transfer factor, which encodes genetic information for transfer between plasmids, and then the R determinant is the gene that's going to confer antibiotic resistance. So a lot of times, if you want a sample DNA or something to be antibiotic resistant, you can put it in a plasmid and transform it into antibiotic resistance. So that's our next topic, transformation. So that's another process leading to genetic recombination, as well as the transfer of genetic information from one bacterium to another. So we have small pieces of extracellular DNA that are taken up by the bacterial cell, and then they get integrated very stably into the chromosome. And it's a two-step process. First is that the foreign DNA enters the recipient cell, and then it recombines between the foreign DNA and the homologous region of the recipient chromosome. And a lot of times too, bacteriophages, we start to see that, but transformation It depends a lot of times on bacteriophages. So they're often bacteriophage is often nicknamed a phage, or it's just shortcut to phages. And phages are bacterial viruses, and so a bacterial virus is really a virus that has a bacteria as its host, and the bacteriophages reproduce via bacterial genetic recombination. So recombination of genetic information, like if you have a bacterial organism and genetic information recombines, oftentimes they can reproduce a bacteriophage. So the structure of the bacteriophage is that it has a head, and against a virus. So the virus or the bacteriophage has a head, and the DNA is contained in a protein coat. And then the structure of the bacteriophage has a tail that contracts within a sheath around it, and it contracts into the central core. And then tail fibers are often present in bacteriophages, and they contain binding sites that recognize the bacterial outer surface, and then they latch onto it, and then they inject their DNA. So here's here's a picture of what it looks like. So here's the little tail right there, and has a collar on it and a tube, and then the sheath right over here, and then these fibers. So the fibers are the little feet that let it sit, and then this tail right there is like a spring. And so this part right there, the head gets pushed down somehow or other, and then that genetic material in there comes out here. So we'll see a picture in a little bit of where this is sitting on a cell and that mechanism of action happens. But you can see here with this micrograph, you can see the head right there, the tail right there, and then these tail fibers that latched onto the surface now. And then this one may look like it's injecting some material out of it. So here's a good example where you have a cell. The phage is absorbed into the bacterial host. So you have your host chromosome right here, and then the phage latches onto the top, and then it makes itself nice and comfy. This part right there plunges down. So the little head right there plunges down, and here's all the material that it lost. It gave up. It injected its DNA. So the phage DNA is injected into the host DNA, and then it does get degraded. It unwound and it's coming apart. But then you can see in number 3, the phage actually got absorbed into the host, and then they mature. They grow into larger phages. Bigger phages, and then they break up the host cells. So they got inside the cell, released its DNA. The DNA let it grow and multiply, and once those phages matured, they now break up their host cell and escape. So notice that it's not a closed circle. It's not a closed circle there. It's an open-ended thing. So it's very linear. We could do that in a straight line. We could show it too. So you get your host cell, and the phage is absorbed to the bacterial host cell, injects its material DNA, and then itself goes inside. All the material gets replicated, and the phage grows, and then it leaves the cell while it's breaking it all up. The plaque assay is actually a technique for determining. It's a lab technique for determining the number of phages that are produced after infecting a bacterial cell. So that's to run an assay, it entails a serial dilution or a whole series of dilutions of virally infected bacteria. And so that's much like those 3 petri dishes we saw, where the slow growing one is actually it's actually a watered down version or diluted version, and it makes counting easier. So after you dilute your infected bacteria here, you want to count the plaques, and their areas clear of bacteria. So you want to count the plaques on plates and determine the number of phages in the original culture. Then lisynergy is another way to transfer genetic information. So here, the phage DNA integrates into the bacterial chromosome, and they coexist together. So the last one, they didn't coexist. The host cell got lysed. But here in lysynergy, no lysis is occurring. It's not ruining its host cell. So with lysynergy, the DNA, the phage DNA just gets replicated along with the chromosome and passed on to the daughter cells, but it never gets broken up. And then you have prophage, which is viral DNA integrated into a bacterial chromosome. And when it's in the lysogenic stage, it's capable of being lysed as a result of the induced viral reproduction. A bacterium harboring a prophage has been lysynergized, as described in the prior slide. And then the viral DNA is classified as an episome, and it's an episome is defined as a genetic molecule that can replicate either in the cytoplasm of a cell or as part of its chromosome. So phages can be mild-mannered, or they can be virulent. So mild-mannered phages are temperate, and they can either lyse cells or behave as a prophage. More often than not, they do not lyse cells. They're often the nicer ones, and they behave as a prophage. But the virulent phages, their only job is to lyse, so they can be really destructive. A virulent phage gets into your cells and starts lysing everything or breaking everything all apart. So to map a bacteriophage, there was a mixed infections experiment where two distinct mutant strains were simultaneously infected in the bacteria, and the viral particles used exceeded the bacterial cells, so that they wanted, and the reason for that was to ensure simultaneous infection. And what they did in this mapping was demonstrated that intergenic or two loci recombinations occurs in bacteriophages. So again, the bacteriophage, a really key concept here is intergenic recombination occurs in bacteriophages. So it's at two different loci. So in sum, our bacteriophages are bacterial viruses that are really safe to humans. They can be manipulated on the genetic and physical level to treat humans with diseases. And the bacteriophages aid in the alteration of genes, and that's what we saw with the little head on it, and it injects its DNA material. And sometimes they just lyse everything all up. Other times they don't. But yeah, bacteriophages can be a source of destruction, but more often than not, it's the good DNA or the good bacteria that's acting. So the bacteriophages aid in the alteration of genes. And an alteration of a special medical significance is the creation of clones, where you have an identical copy of DNA sequence. That's very debatable on, obviously the ethical part of that is very debatable. But we won't, right now we won't go into that. So here's our transduction. And so transduction is bacterial recombination mediated by the bacteriophages. So the bacteriophages played a large role in the last section, and they play a big role here too. So Liederberg and Zinder experiment was using Salmonella, the mutant strains, and it led to the discovery of phage transduction in bacteria. So it's just bacterial, again, transduction is bacterial recombination mediated by bacteriophages. And then co-transduction is two genes are close enough to be transduced simultaneously, or two independent transduction events may occur if they're really not that close. So let's talk about gene mapping. Here's a good one. This gene map, I think it's with the R2, and I forget what that stands for. But I took this out of your textbook, and as I'm flipping through, I'm not finding it right yet. But at any rate, this here is a picture. It's actually a partial map of the mutation in the A and B cistrons at the RLL locus of the T4 phage. So again, to say that here, this is the T4 phage. This is a map, a gene map of the T4 phage, And it also shows an area of key importance. It also shows many mutations right here and many mutations here. A little less mutations there and here, a little bit of mutation there. So this area here, what we end up seeing here with this one is mutations occurring, and then with this one here, their numbering starts at A1A, the beginning, and they're just able to sequentially go all the way around, and then switch over at this part. Cistron B takes over right here, so the numbering changed again. But the key thing again is seeing the mutations, and many mutations occur independently, and but then even when you do get a mutation, assume that it was passed on to you from your parent. Maybe the mutation. Well, actually, that would not be a good assumption. You can it could be a spontaneously generated mutation as well as something inherited or inheritable. So if you have any questions, please do reach out to me.